Before starting any of your processes, you need to ensure that you've undertaken hand decontamination through hand washing, and that you're wearing all the required personal protective equipment that is appropriate for the task you're about to undertake. These will be listed in your standard operating procedures and will include items like lab coats, gloves, goggles, and masks as needed. Once you've finished with the hand wash and put on your PPE, you'll need to prepare your work areas. This includes taking out any of the consumables that will need to be brought to room temperature, as well as cleaning down your workspace. You'll also need to do things like prepare disc pots that contain 10% bleach. Your workspace must be cleaned at the start and end of every day, as well as in between changes of procedure when you'll be working with different specimen types. The same process will also need to be undertaken if there are any spillages. For fluid spillages, you'll need to follow local guidelines, as there may be additional steps that you're required to undertake. Preparing and cleaning down of your workbench includes not just cleaning down the bench space itself, but any of the equipment you're likely to use, including racks and pipettes. This video covers setting up antibiotic sensitivity testing for the pure isolates grown from secondary or purity plates. You need to make sure that the agar plates and antibiotic discs that you will be using have been brought to room temperature and are completely dry. This is to ensure an even bacterial lawn when inoculating. You will be using 2 different types of agar to set up sensitivity testing. For Streptococcus pneumoniae, you'll be using Muller-Hinton agar with 5% defibrinated horse blood and 20 mg beta NAD. And for E. coli, you'll be using Muller-Hinton. It's crucial to use the correct agar in incubation conditions and times when undertaking sensitivity testing, as over or under incubation can impact on the accuracy of readings. You'll set up control plates every time you set up a sensitivity plate in order to help troubleshoot and control for these issues. Now on to the process. Label a suspension tube for each specimen with a number and isolate ID for each isolate to be tested, as well as one for the control strain. This will be ATCC 49619 for Streptococcus pneumoniae, or for E. coli, use ATCC 25922. Make your suspensions of test and control isolates from fresh overnight growth on non-selective medium. This will usually be your secondary plate or TSA plate for E. coli, and for Streptococcus pneumoniae, either a primary or a secondary blood agar plate. Make your suspensions by suspending colonies in saline using either a cotton swab or a sterile disposable loop. When making your suspensions, use 2 to 3 morphologically similar colonies to avoid atypical variants, and mix by inversion. You'll now need to standardise your suspension to a density of Macfarlane 0.5 standard using the Remmel-Macfarlane equivalence turbidity set. In this video we have a different kit available to us, but the principle is the same. Invert the Macfarlane equivalence turbidity set gently to fully suspend the polystyrene particles, and then compare the turbidity of the bacteria suspension prepared. Note: the bacteria suspension tube should be of similar diameter as the Macfarlane equivalence turbidity standard tube. You need to make sure you're doing this in adequate light. Match suspension is when equal obliteration or distortion of the black lines is present. You may need to adjust the turbidity of the suspension by adding either more organism or more saline to achieve the 0.5 Macfarlane standard. It's crucial that the density is correct, as if the inoculum is either too dense or too light, This will alter the zones of inhibition and thus the antibiotic resistance reading. Once the suspension has been made, it must be used within 60 minutes, or a fresh suspension needs to be made, and it's optimal to use it within 15 minutes. If there's any delay post-preparation, the suspension should be mixed by inversion prior to use. Once the suspension is made, you will now progress to inoculating the room temperature agar plates. Plates must be labelled with a permanent marker with the bacterial isolate ID, specimen number, and AMR to indicate that this is the AMR culture plate. The control plates must be labelled with either ATCC 49619, control strain for Streptococcus pneumoniae, or for E. coli use ATCC 25922. Labels can also be printed if this option is available. Now dip a sterile cotton swab into the mixed suspension and remove any excess fluid by turning the swab against the side of the container. This is important to avoid over-inoculation of the plate. Then spread the inoculum evenly across the entire surface of the plate by swabbing in 3 directions. Take particular care to ensure there are no gaps between the streaks. Once this process is complete, you need to repeat it for all isolates to be tested. Once this is done, we will go on and apply the antibiotic discs. Make sure the discs are at room temperature and in date. The disc dispenser should be loaded with the following discs: the Streptococcus pneumoniae, oxacillin, erythromycin, vancomycin, ciprofloxacin, ampicillin, and cotrimoxazole are all to be used. The details of the disc concentration are in the SOP. And then for E. coli, you'll be loading with meropenem, ceftriaxone, ciprofloxacin, ampicillin, cotrim, and gentamicin. The disc concentrations are all in the SOP. Apply the disc within 15 minutes of inoculating the bacterial lawn. This is crucial as the organism may begin to grow and appear falsely resistant or have reduced zone size if plates are left for prolonged periods prior to the application of the discs. Apply the discs as set out in the SOP using an antibiotic disc dispenser. This ensures that the discs are evenly placed across the agar surface, as well as reducing the risk that individual discs become contaminated during placement. There should be no more than 6 discs per plate in order to leave room for complete zones of inhibition to be read and reliably measured. Once the discs are applied, they are incubated within 15 minutes to prevent antibiotics pre-diffusing into the agar, resulting in erroneously enlarged zones of inhibition, i.e. appearing more sensitive than they are. To ensure even heat distribution, stack AMR plates in stacks of no more than 5. E. coli AMR plates should be incubated in a standard incubator at 34 to 36 degrees C in air for 16 to 20 hours, and Streptococcus pneumoniae AMR plates should be incubated in 5% CO2 at 34 to 36 for 16 to 20 hours. Ensure you record all necessary information in the lab book, including the lab ID number, using a secondary printed label if available, the date the AMR plate was streaked, the time placed into the incubator, and any relevant observation or incident. You will then need to clean down your bench space appropriately.